The deeper we plunge into the world of single-cell RNA sequencing, or scRNA-seq, the more intricate it becomes.
Uh-huh.
Think about the incredible potential for, like, pinpointing cancer targets and truly understanding the inner workings of our cells. But there's often a sticking point, right?
Oh, absolutely.
And in this field, a major one has been the really detailed, often manual work of figuring out what kind of cell each one is.
Absolutely. For quite some time, that crucial step, assigning identities to the massive amounts of data coming from scRNA-seq experiments.
Right.
Well, it depended heavily on expert knowledge and, frankly, a lot of manual effort.
Right. Tedious.
It wasn't just slow, it could also introduce some inconsistencies depending on who was doing the annotation.
But get this, the world of large language models, or LLMs, has entered the scene, promising to automate and really accelerate this very process. And what's truly fascinating is that these AI systems are showing a significant level of agreement with the traditional biological tools we've been using.
Yeah, that's the really interesting part.
This suggests they might be picking up on some, fundamental biological signals.
That convergence is really important.
Yeah.
It hints that LLMs aren't just speeding things up, they might be grasping core biological principles in a way that aligns with well-established methods.
Okay.
This could have really exciting implications for how we interpret the data after we've identified the cell types. It's potentially a fundamental shift in how we approach computational biology.
Now, like any new and exciting technology, there are a few things to keep in mind. Actually, getting access to and using these LLMs can be a bit tricky.
For sure.
You've got different companies providing them, each with their own way of doing things, their own APIs.
Right, the interfaces aren't standardized.
And if you're thinking about using the open source models, well, their sheer size and complexity can be pretty demanding in terms of computing power.
Precisely. It's not always straightforward for researchers to just, plug these powerful tools into their current ways of working, their workflows. There's a real need for solutions that can bridge these gaps and make LLM capabilities more readily available.
And that brings us to what we're really digging into today. We've got a new Python package called andIC5-inary.
And dictionary, yeah.
It's a bit of a mouthful, I know, but what it's designed to do sounds pretty groundbreaking. It aims to be completely universal, LLM provider agnostic, as they say.
Right.
Think of it as a universal adapter built on scRNA, which is a real workhorse for scRNA-seq analysis, and using the flexible LangChain framework. The truly amazing part. They say you can connect it to almost any LLM out there with just a single line of code.
That level of simplicity and seamless integration is a real game changer, potentially.
Right.
andIC5-inary looks set to consolidate automated cell type annotation and the inference of biological processes within the familiar scMPy environment. That's a huge step towards making LLMs more accessible to everyone in the field.
It takes away the complexity.
It abstracts away all the complexity of dealing with different LLM APIs. That's a huge win for researchers.
So for this deep dive, you sent us a really interesting set of research. We've got the paper that introduces andIC5-inary.
Yep, the main one.
Another one that rigorously tests a whole range of open source LLMs designed specifically for single-cell data.
The benchmarking study.
And a third looking at the impact of just making LLMs bigger and bigger for this kind of analysis, scaling them up.
That's right, the scaling paper.
Our mission today is to really unpack how LLMs are being used and tested for assigning cell types in scRNA-seq data, with andIC5-inary playing a central role in much of this. Okay, let's get into it, starting with andIC5-inary itself. What's the core idea behind it?
At its heart, andIC5-inary aims to simplify and automate two really important parts of scRNA-seq analysis: figuring out what kind of cell each one is, the annotation part, and understanding the biological processes that are happening within those cells.
Okay.
It does this by providing a single set of tools that works within the scMPy environment, meaning researchers can use LLMs without having to, switch between different platforms or deal with complicated connections.
And it's not just about the basics, right? This package seems to have quite a few smart features built in. Tell us more about what it can do.
Indeed. andIC5-inary tries to automatically figure out the best level of clustering for your data, finding those natural groups of cells. Although the researchers do point out that current LLMs aren't perfect at this just yet, there are limitations.
Still early days for some things.
Right. But where it really shines is in its different ways of assigning cell types. You can do it based on a single list of important genes for each cell type. Marker genes, essentially.
Okay, the standard way.
Or you can compare multiple lists of such genes and even try to figure out more specific subtypes of cells by comparing these lists. And importantly, it can also take into account the specific tissue you're analyzing. It's tissue-aware.
That tissue awareness sounds really key. Because a liver cell isn't the same as a brain cell, obviously.
Exactly. Context matters. What else does it offer? Well, managing the labels we assign to cells is another big part.
The label management.
Yeah. ANDIC5 provides tools to make sure that if you have the same cell type across different data pits, or even within the same one, the labels are consistent, unifying them.
That's huge for comparing studies.
Definitely. It can also help you create a hierarchy of labels, going from very specific subtypes all the way up to broad categories. Like this specific neuron type is part of the broader neuron category.
Right, building a structure.
And perhaps most importantly, it offers ways to use LLMs themselves to see how well different sets of labels agree with each other.
So using the AI to check the AI, in a way.
Sort of, yeah, or to check human labels against AI labels or different AI labels against each other. So it's not just spitting out labels, it's helping you make sense of those labels and how they relate.
Okay, so it's helping you evaluate the output. And what about the genes themselves? Does it do anything there?
ANDIC5 includes functions for working with genes, too. You can add extra information, annotations to sets of genes. Like, for example, you can easily tag all the genes that are involved in, say, the heat shock response, or genes related to a specific pathway.
Gotcha. Functional annotation.
Right. It also has functions that use an LLM to try and infer the biological processes that a given list of genes might be involved in. Like, here's a list of genes upregulated in this cluster. What might they be doing?
Okay, so it's handling both the cells and the genes, all within this one package. And the major selling point is this really simple one-line way to connect to different LLMs. How does that actually work under the hood?
That simplicity comes from building on top of LangChain. And ANDIC5 has a way to set up which LLM you want to use.
Right.
And once you've done that, all of ANDIC5's tools that use an LLM can just call on it without you needing to specify the exact model each time. You just use the configure on backend function once, and then all the LLM-powered parts of ANDIC5 can work with whatever model you've chosen, whether it's from OpenAI, Anthropic, Google, Meta.
The big commercial ones.
Exactly. Or even the ones available through Amazon Bedrock, like Mistral, Titan, and Cohere.
That's pretty powerful. And it sounds like this flexibility was really put to the test on a massive data set, right? You mentioned benchmarking on the Tabula sapiens dataset.
Exactly. The ANDIC5 paper describes a study where they used the package to see how well all the commercially available LLMs could assign cell types to the cells in the Tabula sapiens V2 dataset.
And Tabula sapiens is big.
It's huge. Over 1.2 million cells that have been carefully annotated by human experts. But what's particularly interesting is that when they did this study, more than half of those expert annotations hadn't been made publicly available yet.
Oh, wow.
Meaning the LLMs hadn't been directly trained on that specific information.
So it's a real test of their general biological knowledge, not just pattern matching on the training data.
Exactly. A genuine test.
So how did they actually run this test? What were the steps they took?
For each tissue in the Tabula sapiens dataset, they followed a pretty standard set of steps for analyzing scRNA-seq data. First, they normalized the data and applied a log transformation. Standard stuff. Then, they selected the 2,000 most variable genes.
Focusing on the informative ones.
Right. Scaled the data, performed principal component analysis, or PCA, to reduce the complexity, calculated a neighborhood graph to see which cells are similar to each other, and finally used UMB to visualize the different cell populations in a more easily understandable way. These PCA and UMP methods just help make the complex data manageable and viewable.
Standard practice for scRNA-seq. Okay. And then the LLM came in to assign the labels.
Precisely. They used a two-step process. First, each group of similar cells, the clusters identified in the previous steps, was independently annotated using an LLM based on its top 10 most distinctive genes, its marker genes.
Okay, step 1. Initial annotation per cluster.
Then, in the second step, they used an LLM again to merge any labels that were essentially the same but written slightly differently, things like capitalization or minor variations in terminology.
Cleaning up the labels.
Exactly. And also to correct any isolated instances where a cell might have been assigned a very specific subtype that didn't quite fit with the rest of its cluster, kind of smoothing things out.
Makes sense.
They decided to focus on the agreement at a more general cell type level because they found that consistently getting the LLMs to identify very fine-grained subtypes was challenging, less reliable.
That makes sense. getting consistent agreement at the broader level is probably more important for the initial overall understanding anyway. So who were the top performers in this LLM competition? Which ones agreed most often with the human experts?
According to their findings, Claude 3.5 Sonnet showed the highest level of agreement with the manual annotations when looking at individual cells. It got over 81% of them right in a binary sense, like, is this the right broad category?
Okay, 81%. Pretty good.
Claude 3 Opus, on the other hand, had the highest agreement at the level of cell types, around 65%, and also had the highest proportion of perfect matches, both for individual cells, over 71%, and for the cell types themselves, over 52%.
So Opus was maybe getting the exact label right more often.
That seems to be the case, yeah. Other models that performed well included Gemini 1.5 Pro and GPT-4o, the big players.
Those are some of the biggest names in the LLM world, yeah. Were there any that didn't do so well? Any surprises?
Yes, the study noted that the smaller, more lightweight models, those with fewer parameters, generally didn't perform as well.
Oh.
Which perhaps isn't too surprising.
Less capacity, maybe.
Possibly. Interestingly, they also found that Amazon's Titan models weren't reliable enough at following instructions to be effectively used for this specific cell type annotation task.
Ah.
Yeah, so they weren't included in the main comparison results because they just couldn't consistently generate the annotations in the required format.
That's a really useful practical takeaway. So some models just aren't suited for this kind of structured output yet.
Seems so.
And it wasn't just about how well they agreed with the human labels, was it? They also looked at how well the LLMs agreed with each other.
Exactly. They used a statistical measure called kappa to assess the inter-LLM agreement. A higher kappa value means the AI systems are in more agreement, either with each other or with the human experts.
Okay, so who agreed most?
Claude 3 Opus showed the highest consistency with the manual annotations, having the highest kappa value there. GPT-4o had the highest average kappa value when compared to all the other LLMs, indicating good overall consistency across the board.
Interesting.
And the pair that showed the most consistent annotations with each other were Claude 3 Opus and Claude 3.5 Sonnet. They agreed really well.
Fascinating. So the LLMs that performed best seem to be converging on similar answers, which definitely adds confidence to their potential, right?
It does suggest a level of reliability, yes.
Now, looking more closely at the specific cell types, where did the LLMs do really well, and where did they struggle a bit in their annotations?
The study found that LLMs generally showed strong performance in annotating the major, more common cell types. We're talking over 80–90% agreement for the 10 most abundant cell types in the Tabula sapiens dataset.
Okay, so good on the big populations.
Mostly, yes. However, there were a couple of notable exceptions. Stromal cells and basal cells. They seem trickier.
Tell us more about where they had trouble. What happened with those cell types? Basal cells first.
For basal cells, the top-performing LLMs often misclassify them as epithelial cells.
Why might that be?
Well, it isn't entirely surprising because these two cell types are actually closely related developmentally. They share a lineage.
Ah, okay.
And based on a small set of well-known marker genes, their expression patterns might overlap somewhat. Some basal cells might express KRT8 or EPGAM, which are more epithelial, while lacking strong KRT5 or TP63, the classic basal markers.
So it's not necessarily a complete mistake, but more of a challenge in making a finer distinction based on maybe ambiguous markers.
Precisely. The LMs seem to be picking up on shared characteristics, or maybe the markers weren't perfectly specific. It highlights biological nuance.
Okay, and what about the stromal cells?
The case of stromal cells was a bit different. These cells in the Tabula sapiens dataset mainly came from the ovaries.
Right.
Now, genes that are generally known to be markers of stromal cells, like DCN or LUM, were expressed across these manually annotated cells. But the LLMs tended to assign more specific subcluster labels within this population.
So the manual label was broad stromal, but the LLMs were saying this kind of stromal and that kind of stromal.
Kinda, yeah. This could suggest a couple of things. Maybe the initial clustering of the data was too broad, lumping different stromal subtypes together. Or perhaps the chosen marker genes didn't fully capture the diversity within the stromal cell population as the LMMs perceived it based on the full gene list.
Interesting. So the LLMs might be picking up on more subtle differences that the broader manual label doesn't fully capture in that specific tissue context.
It's a possibility. It points to the complexity of defining cell states versus stable types.
Were there any other consistent misclassifications mentioned?
Yes. One other notable observation mentioned in the supplementary figures was that cells manually annotated as neutrophils were frequently labeled as macrophages by the LLMs.
Neutrophils and macrophages, immune cells, often tricky.
Exactly. This points to a potential difficulty in distinguishing these two types of immune cells based solely on a limited number of differentially expressed marker genes, which is a known challenge in the field, even for manual annotation sometimes.
That really highlights how important it is to carefully choose the marker genes we use for annotation, doesn't it? Or maybe use more complex methods.
It certainly does. Or maybe rely more on the LLM's ability to synthesize info from many genes.
What about how well the different LLMs agreed with each other across all the various cell types? Was it generally high?
The study found that for nearly all cell types, there was more than 50% agreement between the different LLMs.
Okay, so better than chance, suggesting they aren't just guessing wildly.
Right. It suggests low rates of completely spurious random annotation. They also noticed a trend. Major cell types tended to have high agreement both between the LLMs and with the manual annotations.
That makes sense.
While smaller, less common cell types often had moderate agreement among LLMs, but lower agreement with the expert labels.
Right. Less data, maybe less consensus in the literature for the rarer types.
That makes intuitive sense. The more data and established knowledge there is about a cell type, the more likely everyone, including the AIs, is to agree on its identity.
And you mentioned one specific case where the LLM's label might actually be more pragmatic than the manual one, mononuclear phagocytes.
Yes, that was regarding the mononuclear phagocytes. The manual annotation identified these cells as such, which is a broad category including monocytes, macrophages, and dendritic cells.
Right.
But the top-performing LLM consistently labeled them simply as macrophages.
Okay. So was the LLM wrong or just less precise?
Well, when they looked more closely at the expression of key marker genes, there was evidence to suggest that this cluster of cells did indeed have a strong macrophage signature, even though it likely also contained monocytes and dendritic cells.
Yeah, I see.
The researchers suggest that while the manual label mononuclear phagocyte might be technically correct as a broader category, the LLM's macrophage label could be more representative of the dominant characteristics within that particular cluster based on the overall gene expression pattern.
So it's a difference in annotation depth or interpretation, perhaps.
Right.
The LLM focusing on the most prominent signal.
Exactly. It really highlights that how we define and categorize cell types can sometimes have different levels of detail or interpretation. It also raises questions about potential over-clustering in the initial analysis or limitations in just using the top 10 marker genes.
It brings up an important point about the nuances of cell type definition and how different approaches might prioritize different levels of granularity. Fascinating stuff. Now, shifting focus a bit, the second paper you shared looked at how well open source LLMs designed specifically for single cell data perform. What were the main findings there?
Right. This study took a really close look at the performance of 5 specific open source LLMs. These are models developed for single-cell analysis. Their names are scGPT, Geniformer, cBERT, CellMM, and tGPT.
Okay, specialized models.
Yeah. They tested them across a wide range of 8 different tasks, not just annotation, using 22 different datasets. It's important to note, though, that not every model was tested on every single dataset for various reasons outlined in the paper.
Makes sense.
And importantly, they also compared these models to state-of-the-art methods that are specifically designed for each of those individual tasks. Like, how does scGPT compare to a dedicated clustering algorithm?
So a real head-to-head comparison. What did they find when they focused specifically on the task of assigning cell types, the annotation task?
For cell type annotation, they found that, on average, the models that had been pre-trained on large amounts of single-cell data generally performed better than those that hadn't. Pre-training seemed to help.
Okay, that tracks. Prior knowledge is good.
But there was a fair amount of variability between the different models. No single model totally dominated. Interestingly, when it came to predicting cell types within the same dataset, the model had been trained on what they call intra-dataset prediction. All of the single-cell LLMs showed pretty comparable performance, even if they had different pre-training methods or architectures.
That suggests that even without extensive pre-training, these models can learn to distinguish cell types within a given experimental context fairly well.
It seems so, yes. They can adapt to the specific dataset.
Yeah.
What about how reliable and consistent they were? Were there issues?
Yeah, how robust were they?
There were noticeable differences in how well the different open-source LLMs performed across datasets. The researchers specifically mentioned issues with the reliability of CellM.
CellM, what kind of issues?
It apparently encountered running errors on several datasets, which raises concerns about how easily it could be used in different situations or by different labs. Practical usability issues.
Right, if it keeps crashing, it's not very helpful.
Exactly. The overall conclusion was that single-cell LLMs can be effective at cell type annotation, provided they have been trained on appropriate data and have a suitable model structure. Makes sense.
What?
But they emphasized that no single open-source framework stood out as being consistently the best across all the datasets they examined.
Ah.
So while promising, the landscape of open-source single-cell LLMs is still evolving, and there isn't yet a clear winner for all scenarios. You might need to try a few depending on your specific data and task.
So more tools in the toolbox, but no silver bullet yet for the open source options.
Okay.
Now, the third paper in our collection took an even broader perspective, looking at the impact of just making general-purpose LLMs bigger and bigger for single-cell analysis. What's the main idea there?
This work builds on something called the cell-to-sentence, or C2S framework.
Cell-to-sentence.
Yeah. The basic idea of C2S is to take the gene expression profile of each individual cell and represent it as a textual cell sentence.
How does that work?
Think of each gene as a word, and the level of its expression, like its count, as how often that word appears in the sentence. So a cell with high expression of gene A and low expression of gene B might have a sentence with many A's and few B's.
Okay, that's a really clever way to translate biological data into something a language model can understand.
Exactly. It allows researchers to use the power of natural language processing techniques, which are incredibly advanced now, on single-cell data.
So what did they do with these cell sentences? Did they just feed them into standard LLMs?
Pretty much, but on a massive scale. They trained very large language models, we're talking about models, with up to 27 billion parameters on a huge collection of over 1 billion tokens.
Wow.
And this collection wasn't just made up of these transcriptomic cell sentences, but also included a vast amount of biological text, like from research papers, textbooks, and other relevant information, metadata.
So mixing gene data with actual biological knowledge.
Exactly. And the researchers found that by making the model this large and feeding it this diverse data, they saw consistent improvements, not only in its ability to predict and generate information, but also in its ability to handle more complex tasks, things that require understanding relationships between many cells and different biological contexts.
So in this case, it really does seem like bigger can be better, at least up to a point.
The scaling seems to yield benefits, yes. And they're not just relying on sheer size, are they? You mentioned they're also refining how they train these massive models.
How so?
Absolutely. They used very specific and targeted ways of fine-tuning the models, often using techniques called reinforcement learning to really optimize them for particular biological tasks.
Okay, so tuning them for specific jobs after the initial huge training.
Right. This allowed them to achieve excellent results in areas like predicting how cells will respond to different treatments or perturbations, understanding natural language related to biology, and even performing more complex biological reasoning.
The ultimate goal here seems to be something more than just identifying cell types or predicting responses, right? They mentioned the idea of virtual cells. What does that mean? That sounds futuristic.
It does. The long-term vision is that by bringing together transcriptomic data and biological text at this massive scale, these very large LLMs could potentially become even better at understanding biology than either specialized single-cell models or general-purpose LLMs are on their own.
A unified model.
Kind of. Leading to the creation of these virtual cells, essentially very sophisticated computer models that can accurately represent and even predict how real cells will behave in various situations. Imagine being able to simulate a cell's response to a new drug entirely in silico.
Yeah, that's profound. The potential for virtual cells to revolutionize drug discovery or our understanding of disease progression is huge.
It's a truly profound potential insight from this research, yeah. A long-term goal, but very exciting.
That's a pretty mind-blowing concept. It sounds like we're heading towards a future where AI can not only analyze single-cell data, but also help us simulate and deeply understand cellular behavior. Okay, now, to round out our understanding of the AI architectures involved here, you also included a survey paper on transformer-based models in single-cell analysis. For those of us who aren't, deep experts in machine learning, what are the key things to understand about transformers in this context?
Okay, yeah. Think of the transformer architecture as a really powerful and flexible engine for processing sequences of data.
Sequences like text or music or...
Or, in the context of single-cell analysis, that sequence can be thought of as the list of genes that are expressed in a cell, maybe ordered in some way, or just treated as a set.
Got it. A list of genes and their expression levels.
Right. The architecture typically includes parts called encoders that learn to understand the input data, the gene list, and decoders that can generate output, like a cell type label or a predicted response.
Encoder, decoder.
Key components include things like multi-head attention mechanisms.
Attention. Like paying attention.
Exactly like that, in a computational sense. It allows the model to weigh the importance of different genes or gene combinations when making a prediction, rather than just looking at them one by one. It can pay attention to multiple relationships simultaneously.
Oh yeah, that sounds powerful.
It is. And there are also techniques called residual connections and layer normalization that basically help these very deep, complex networks train effectively without losing information. So it's a sophisticated way for the AI to weigh the importance of different genes and their relationships within a cell.
So it's this attention mechanism that's really key to understanding gene relationships. You also mentioned SBERT as a particularly important early example of using transformers.
Precisely. And this transformer architecture has been adapted and used in various models for scRNA-seq analysis, not just the huge LLMs. For example, models like transclusters, transort, CI-form, STGRNS, and TGEM all use transformers for tasks like predicting cell types. Some even incorporate additional methods like linear discriminant analysis or CNNs, or ways to understand patterns in gene expression motifs to try and improve performance and make the results more interpretable. Explainable AI is important here too.
Right, understanding why it made a prediction.
Yes, and scRNA-BERT was one of the first pre-trained single-cell models that was built on the BERT architecture, which is a specific, very influential type of transformer originally for language.
Okay.
A key innovation in scRNA-BERT was the use of something called GNA2VEC. It's a way for the AI to learn numerical representations or embeddings for genes based on which other genes they often appear with or have similar roles, like creating a dictionary of gene relationships beforehand.
So it learns gene context first.
Kinda, yeah. It then uses this gene embedding information along with the actual gene expression data as input for the transformer. And to handle the very large number of genes in scRNA-seq data, thousands, right? scRNA-BERT uses a clever trick called the performer mechanism. It's a way to approximate the computationally expensive self-attention calculation, allowing it to process data with over 16,000 genes without becoming overwhelmed, making it feasible.
Gotcha. Efficiency matters.
Definitely. And these transformer-based models, both the big LLMs and specialized ones like scRNA-BERT, have been used for a wide variety of tasks beyond just cell type annotation. Things like correcting for technical differences between batches of data, grouping cells into clusters, figuring out gene regulatory networks, and predicting how cells will respond to perturbations or treatments.
It's clear that the transformer architecture is a fundamental building block for these advanced AI tools in single-cell biology. Really versatile.
Absolutely. It's proven very effective.
Well, we've covered a huge amount of ground in this deep dive. This has been really illuminating. To bring it all together, what are the key insights you'd want our listeners to take away from all of this?
I think the main takeaway is that large language models are significantly changing how we automate and potentially improve the accuracy of cell type annotation in single-cell RNA sequencing. That's clear. And crucially, they're becoming more accessible, especially when they're integrated into familiar workflows through tools like Covenary.
Making it easier for biologists to actually use them.
Right. Our look at the results for the Tabula sapiens dataset shows that commercial models like Claude 3.5 Sonnet and Claude 3 Opus are currently achieving really impressive results on large, complex datasets. They're setting a high bar.
But the open source world isn't standing still?
Not at all. The research on open source single-cell LLMs and the work on scaling up general-purpose LLMs for this field also show a lot of promise and ongoing progress across a whole range of single-cell analysis tasks. It's a very active area.
And while the progress is undeniable, it sounds like there are still some challenges to overcome. It's not perfect yet.
Absolutely. We still see challenges in consistently annotating certain cell types, such as those basal and stromal cells we discussed. That highlights the ongoing need to think carefully about how we define cell types, how we select markers, and to further refine both LLM technology and our fundamental understanding of cell biology.
Right. The biology and the tech have to advance together.
Exactly. And the fact that different open source models perform differently and some have reliability issues also shows that this is a field that is still very much evolving. It's not mature yet.
But the development of tools like Cavenary seems really important for navigating this evolving landscape, a kind of central hub.
Exactly. Cavenary provides a flexible and unified way for researchers to easily harness the power of different LLMs, commercial or maybe eventually open source ones too, without getting bogged down in the technical details of making them work together. This ease of use is crucial for wider adoption and further development.
Lowering the barrier to entry. And looking to the future, what's the big picture? Where do you see this field heading? Is it all about virtual cells?
I think the future of single-cell analysis is definitely linked to the continued growth in the size and sophistication of LLMs, for sure, but also the development of frameworks that can seamlessly integrate different types of biological data, not just RNA, but maybe proteomics, epigenomics, spatial data. Multiomics, exactly.
Yeah.
And integrating all that with the vast amount of text-based biological knowledge we already have in papers and databases. The idea of virtual cells, these sophisticated AI models that can predict cellular behavior, that represents a really exciting frontier that could significantly deepen our understanding of biology.
It really could. This has been truly fascinating. As these AI models become more deeply integrated into biological research, it really makes you wonder, how will our fundamental understanding of what defines a cell type and how cells function actually change? Will the definitions become more fluid, more context-dependent?
That's a great question. It might push us towards more dynamic definitions.
And what completely new questions will these powerful tools enable us to even ask? Questions we couldn't conceive of before. For those of you who want to explore this rapidly advancing field in more detail, we've included links to the Andic-5 GitHub repository and a cell type annotation leaderboard in our resources.
Definitely worth checking out if you're interested.
It's a field that's moving incredibly quickly, and it's definitely one to keep an eye on. Thanks for taking this deep dive with us.